Hello everyone, I'm myself Dr Dipti and today we'll discuss about synapse. Now, first of all, what is synapse? Synapse, it is the site where nerve cells they communicate with each other. Here you can see this is the site of synapse. The term synapse was first introduced by Sherrington in 1924. In 1924, okay, and it is defined as a site of physiological continuity but not anatomical continuation. Okay? So you can see here two neurons for functional purpose of use they come together and they come in contact with each other for the transmission of impulse. So there is no anatomical continuation but functional continuation is there between two neurons. Okay? In CNS we have about 10 raised to 11 neurons and 2 into 10 raised to 14 synapses are present. Okay, now, in each synapse, you have at least two neurons. One that is presynaptic neuron or presynaptic terminal. Okay, it is the terminal or the neuron which carries information towards synapse, this one. And other is postsynaptic neuron or postsynaptic terminal, this one, this is. It carries information away from the synapse. Okay. Sometimes one neuron which is postsynaptic for this synapse, it may be presynaptic for other one. Now, classification of synapse. There are two methods of the classification. One that is anatomical classification and other is physiological or functional classification. Anatomical classification, the synapses are of four types. Exodendritic synapses, you can see here, this one. Exosomatic synapses, exoexonic synapses and dendritodendritic synapses. We'll discuss one by one. First, that is exodendritic synapses. They are most common type of synapses in brain, in central nervous system. Here, there is synapse between axon of presynaptic terminal, this one, and dendrite of postsynaptic terminal. And this synapses are present in motor neurons of the spinal cord. So they are located in the spinal cord. Synapses in the cerebral cortex and in the cerebellum, climbing fiber of the cerebellum. These are the sites where exodendritic type of synapses are present. Second variety of synapses are exosomatic, means here presynaptic neuron, axon of presynaptic neuron synapse with the soma of the postsynaptic neuron here, and they are located in again in the spinal cord, motor neurons of the spinal cord. Some of the motor neurons have exosomatic type of synapses. Okay, then cerebellum, basket cells of the cerebellum, and autonomic ganglia. Autonomic ganglia. Then third variety, that is axonal synapse, axon of one neuron or presynaptic neuron, synapse with the axon of the postsynaptic neuron, and they are found in spinal cord. Okay. Last variety, they are dendritodendritic synapses. They are least common and they are present here synapse between dendrites of first and dendrites of second neuron, and these are present in the mitral and granule cells in olfactory bulb. Olfactory bulb. Okay, these are dendritodendritic synapses. Now important thing is most of the synapses they have axonous presynaptic terminal. Why? Because information is carried out away from the cell body by axon. So when information is carried out away from the cell body, that can be carried out towards the synapse. So in most of the synapses, presynaptic neuron, that is axon part of the neuron. Now, physiological classification, two varieties of synapses are there. One that is electrical synapse and other is chemical synapse. Electrical synapses, here, there is gap junction between pre and postsynaptic neuron. So, information will directly pass from presynaptic to postsynaptic. Whereas in chemical synapses, this information will pass in the form of action potential from presynaptic. Then there is release of chemical neurotransmitter and which helps in the transmission of action potential. Now, what are the characteristics of electrical synapses? Here, as we have discussed, there are gap junctions. So, there is, you can say, physiological continuation is there. So, synaptic transmission is through gap junction and that is fast. So, you can say that synaptic delay, that is less, or maybe no synaptic delay. Okay. Then, here transmission is in both the direction. Both the direction means impulse will pass from presynaptic to postsynaptic as well as postsynaptic to presynaptic neuron. Okay, so that is another characteristic of electrical synapse. Third characteristic that is present in both electrical as well as chemical synapses, that is they both are sensitive to hypoxia. If there is hypoxia, synaptic transmission is affected. Okay. This variety of synapses are not commonly seen in central nervous system. Some of the neurons in the central nervous system, like in some neurons of retina and olfactory bulb, we have electrical synapses. They also found commonly in the lower invertebrates and invertebrates and lower vertebrates. Also, they are present in the muscles of the smooth muscle in intestine and epithelial cells of the lens of eye and cardiac muscle cell. Sometimes they are also present in vestibular nucleus, lateral vestibular nucleus, in the medulla, hippocampus, and some of the neurons of cerebral cortex. Now, second variety of synapses are chemical synapses. Here, the impulse is transmitted by the release of neurotransmitter, that is chemical. So, these synapses are known as chemical synapses. Common synapses in the central nervous system, they are chemical synapses. So, here you can see the structure is similar to neuromuscular junction. There is no continuity. Okay, pre and post synaptic neurons are not in contact with each other. So, therefore, here synaptic delay is more as compared to electrical synapse. That is one characteristic, synaptic delay. Second characteristic of the chemical synapse, that is unidirectional transmission. In one direction only impulses are transmitted. And third, that is hypoxia, they are also sensitive to hypoxia. Large number of neurons in the central nervous system, they are having chemical synapses. Now here, which events they take place when there is synaptic transmission? So first of all, there is arrival of action potential. Action potential comes at presynaptic neuron. Then there is opening of voltage gated calcium channels, they open. And when calcium ions they enter inside, uh, they cause movement of the vesicles near the membrane, and these vesicles they burst, and there is exocytosis of the neurotransmitter. Neurotransmitter comes out. Okay, this neurotransmitter, it passes through synaptic cleft, it binds with the channels which are present on the postsynaptic membrane here. These are known as ligand gated channels. So when this ligand in the form of neurotransmitter binds with the channel, it causes opening of the channel, and there is release of entry of sodium ions. When the neurotransmitter is acetylcholine, so you can say when sodium ions enter, membrane potential that gets depolarized state. So this is the action potential which was here. Now it gets transmitted to postsynaptic membrane. Okay. After neurotransmitter completes its action, it is degraded by the enzyme which is present in the synaptic cleft. Here for acetylcholine, we have acetylcholine esterase present. Okay. Now, one more important thing here. When one action potential comes, about 300 vesicles they rupture. 300 vesicles, and each vesicle is having about 3,000 molecules of acetylcholine or neurotransmitter, any other neurotransmitter. Now we'll discuss about functional anatomy of chemical synapses. You can see here, first as we have discussed, there is presynaptic neuron which carries information towards synapse, and postsynaptic neuron carries information away from the synapse. Okay. So first of all, when presynaptic neuron, it participates in the formation of synapse. Here you can see there is this presynaptic neuron leaves its myelin sheath. Suppose this is the myelin sheath. I'm changing the color so it becomes more clear for you. You can see here this is the myelin sheath. Now first of all, this myelin sheath is left. Now this presynaptic axon terminal, it forms a swelling that is known as synaptic knob, or it is also known as synaptic button. Number of synaptic node or button that varies in depending on the type of synapse where it is located. Now this we have discussed. This synaptic knobs or button, they are of two types. Number one, that is terminal knobs, or second variety, they are coil knobs. So here terminal knobs, you can see here in this diagram, terminal knobs are visible. So when the terminal knobs are there, these are concerned with the excitatory function. Terminal knobs are having neurotransmitter which is having excitation, so excitatory function, right? And terminal coils, it is like a coil like this, so they are concerned with inhibitory function. Inhibitory neurotransmitter is present, that is to be found out by the studies. Now another is presynaptic terminal is also having mitochondria which produces energy that is required for the synthesis of neurotransmitter. It is also having Golgi apparatus. Golgi apparatus is required for processing and packaging of this neurotransmitter. Synaptic vesicles are present which is having neurotransmitter. The diameter of synaptic vesicle is about 50 nanometer, and it contains packets of neurotransmitter which is required for excitation as well as inhibition of the synapse. This neurotransmitter is either excitatory or inhibitory. Now these vesicles, they are produced from the soma, and this vesicle gets transported by exoplasmic flow through exoplasm, that is the cytoplasm in the axon, and that reaches to the presynaptic terminal. Now this we have discussed. Now different type of vesicles are there and they have different neurotransmitter. If the vesicle is small and clear, neurotransmitter may be acetylcholine, glycine, glutamate, and gamma amino butyric acid. If the vesicle is small and dense, neurotransmitter is catecholamine. Large and dense neurotransmitter is neuropeptides. Okay. Now, synaptic cleft, as it is a gap between presynaptic and postsynaptic neuron, the gap is about 20 to 40 nanometer. And it is having extracellular fluid, and it is having the enzyme which causes degradation of the neurotransmitter. For example, for acetylcholine, there is acetylcholine esterase enzyme is available. Okay. Now, postsynaptic membrane has got receptors. There are two kinds of receptors present. One, that is ion channel receptor protein, means when this ligand binds with this channel, that causes opening of the ion channel, and now there is entry of ions. And second is enzyme type receptor protein. When neurotransmitter binds with this ligand, that causes activation of the enzyme, like enzyme for the genes, like enzyme for the protein kinases. So in that way also this transmission takes place. Now after completion of the action, as we have discussed, neurotransmitter is degraded. For acetylcholine, there is acetylcholine esterase present, and for norepinephrine, there is monoamine oxidase that is available, which causes inactivation of the neurotransmitter. Okay. Now, what are the functions of synapses? Synapse, the name itself suggests that is for transmission, synaptic transmission, transmission of the impulse or action potential that is transmitted from presynaptic to postsynaptic neurons. So two types of synapses are there. Those who are concerned with the excitation or transmission of impulse, they are known as excitatory synapses, and those they prevent transmission, they are inhibitory synapses. Now, so here excitatory function, as we have already discussed, when presynaptic axon terminal is having action potential, arrival of action potential here. Okay, then opening of calcium channel, entry of calcium, release of neurotransmitter, and opening of sodium channel. Now very important terminology here is EPSP, excitatory postsynaptic potential. Now what is this excitatory and inhibitory postsynaptic potential? So here, this is excitatory postsynaptic potential when a stimulus is given that is less than threshold. Threshold, or that is, you can see here, it is also known as minimal stimulus, minimal or liminal or threshold stimulus. When the stimulus is less than threshold, when the stimulus is threshold, there is generation of action potential. But if the stimulus is less than threshold, then there is generation of graded potential. Okay. And when the graded potential produced at synapse, that is known as excitatory postsynaptic potential. Okay. So you can see here, whenever the stimulus is less than threshold, this is the threshold one. So when it is less than threshold, what happens? There is generation of non-propagated, means it will not pass, it remains here only. Action potential always propagates, means action potential is generated here, then it will pass such a way. But here it is non-propagated. Graded means it can be added, summated. Suppose EPSP, first of all EPSP, that is 5 millivolt, then again more 5 millivolts, so both are added. And it is monophasic, means it is just one phase, depolarization or hyperpolarization. This is coming back to normal, no other phase is there, and it doesn't follow all or none law. Okay, so this depolarizing response, it starts after zero 0.5 millisecond, it rises to peak within 1 to 1.5 millisecond, and then it comes back within 4 millisecond. The magnitude is about 8 millivolt. Okay, so suppose it is minus 70 resting membrane potential, it may be 8 plus, that is about your 62 millivolt, minus 62, it may not reach to the threshold. Now, ionic basis of the EPSP, first we'll discuss ionic basis, that is because it is excitatory, so there is release or there is opening of, there is release of the neurotransmitter which causes opening of either sodium or calcium channel. So that results in entry of the sodium or calcium. And here most common excitatory neurotransmitters, they are your glutamate or acetylcholine. Okay. Properties we have already discussed. One more property that we have forgotten, that is it is having it does not have refractory period. Does not have refractory period. Refractory period is the property of action potential. Fine. So these are the properties: non-propagated, graded, or you can say summated. This is same thing. No threshold, non-threshold stimulus, does not obey all or none law, or no refractory period. Okay. Now, okay. Then, but one more thing here is, if your EPSP, if your EPSP is strong enough, then it causes opening of voltage gated channels, sodium channels, and that causes depolarization. We will discuss. I think it is here, this one. You can see here, when your EPSP is here, this is this one, then again next EPSP, then again third EPSP, so they all are summated, and when this summation reaches to threshold, then it results in generation of action potential. So you can see here that is summation. Okay. Next is IPSP. IPSP, that is inhibitory, the name itself suggests, inhibitory postsynaptic potential. Here there is hyperpolarization. You can see here there is hyperpolarization seen. So this is hyperpolarizing response. Okay, this response starts with a latency of 1 to 1.5 milliseconds, lower than EPSP, reaches to peak by 1.5 to 2 milliseconds, and it comes back to normal within 3 milliseconds. And here there is release of inhibitory neurotransmitter. Most common inhibitory neurotransmitter, that is gamma amino butyric acid. Okay. Now, properties of IPSP, here also it is because of subthreshold stimulus, not the threshold. It may not reach to threshold. There is always hyperpolarization when there is inhibitory potential, and it changes the membrane potential. More negative increases, but that becomes more negative, and it also can summit, and it has no refractory period. It doesn't follow all or none law. So these are all the properties of inhibitory potential. Now, inhibition of the synapse, that is classified in different 4 ways. Number 1 is postsynaptic inhibition, presynaptic inhibition, Rancho cell inhibition, and feed forward inhibition. We will discuss one by one. So first is postsynaptic inhibition. Postsynaptic inhibition is again of 2 types, direct and indirect. Now, what is direct postsynaptic inhibition? That is, which we have already discussed. Whenever the neurotransmitter release, that is inhibitory, like gamma amino butyric acid, what happens? There is opening of potassium channel or chloride channel. So when potassium channel opens, what happens? Because suppose this is your cell, this is negative inside, positive outside, this is resting membrane potential. Okay. When sodium channels open, because sodium is more outside, so sodium ions, they enter inside, so there is depolarization. Membrane potential, instead of negative, now it becomes positive. But when there is opening of potassium channel, because potassium is intracellular cation, so now potassium, they move out, and when potassium moves out, here the resting membrane potential is -70. Now more positive ions, they are moving out, so the membrane potential goes down. So this is the basis of EPSP, sorry, IPSP. Second is chloride ions, because chloride is again outside. So when chloride channels, they open, there is entry of chloride. So always whenever channels, they open, there is passive transport of ions. Ions, they enter inside, because chloride ions are more outside. Okay, so and when negative ions, they enter inside, again there is hyperpolarization. So this is known as direct postsynaptic inhibition 1. Second is indirect. Here what happens? Suppose this is one synapse. Okay, here one action potential has passed, and one action potential when has passed, then the synapse is in refractory period. And when the synapse is in refractory period, and at that time, if some other stimulus comes, then that stimulus cannot stimulate this synapse, so you can say that it will be inhibited. This is known as indirect inhibition. Okay, so this is postsynaptic inhibition, means postsynaptic membrane is inhibited. Then next is presynaptic inhibition. Here you can see this presynaptic release of neurotransmitter is inhibited. Here you can see, suppose this is the action potential which passes, but some inhibitory neuron is there which inhibits release of neurotransmitter. So it decreases release of neurotransmitter from presynaptic terminal. So this is known as presynaptic inhibition. Presynaptic terminal will release less amount of neurotransmitter. Suppose it is acetylcholine, but if it is inhibited by here GABA, then less amount of acetylcholine is released. So you can say presynaptic terminal is inhibited. Then Rancho cell inhibition. Rancho cells are the inhibitory interneurons. These are the inhibitory neurons in excitatory circuit. Means you can see here. This is one excitatory neuron. Okay, then it stimulates some other neuron, but this neuron is going to stimulate Rancho cell. Now Rancho cell inhibits the same neuron. Means suppose neuron number 1 stimulates neuron number 2, neuron number 2 stimulates Rancho cell, and Rancho cell themselves they inhibit neuron number 2. So this is known as Rancho cell inhibition. Rancho cells, they are inhibitory interneurons, they inhibit in between. Then feed forward inhibition. Here what happens, suppose your number 1 neuron stimulates number 2 and number 3. I will explain with example also, okay? But your 2 is inhibit number 3. This is the circuit. You can see here number 1 stimulates both 2 and 3, but 2 stimulates 2 inhibits number 3. I repeat, one number neuron stimulates number 2 and 3, but 2 inhibits 3. So what happens when 1 is stimulated, 2 and 3 both are stimulated, but 3 is not stimulated much because it is inhibited by number 2. This is feed forward inhibition. This is seen in the cerebellum. In cerebellum, suppose granule cells, when they are stimulated, granule cells, they stimulate basket cell and Purkinje cell both. But basket cells, they inhibit Purkinje cells. So Purkinje cell activity is kept in check by basket cell. Okay, so you can see, suppose granule cells I am going to stimulate. So my basket cell and Purkinje cells both are stimulated, but basket cell stimulation is more than Purkinje because this basket one, they inhibit Purkinje cell. So this is feed forward inhibition. So we have discussed 4 types of inhibition. Number 1, that is post synaptic inhibition. That is again direct and indirect. Presynaptic inhibition, Rancho cell inhibition, and feed forward inhibition. Okay, so these are different varieties of inhibition. Now, what is the significance of synaptic inhibition? Because of inhibition, the activity that is restricted. So synaptic transmission that is kept in check, and that helps to select exact number of neurons to stimulate and cut the extra impulses. And what is applied aspect related with this inhibition is, suppose some drug, that is strychnine. Strychnine destroys inhibitory system. So when this inhibition is destroyed, that results in continuous activity that may result in convergence. So strychnine poisoning, the person is having conversion because of lack of inhibition. Same way in Parkinson's disease, because dopamine, that is inhibitory neurotransmitter, that is impaired. So that produces rigidity, that produces tremor and all. So the synaptic inhibition is also important. So synapse has mainly 2 functions, excitation and inhibition. Okay, so this is all about synapse. You will find all my notes in the soft copy, and the link for the same that is given in the description box. Thank you so much.